reason his slides are reversed. Can we fix that with the camera? Yeah, it's showing mirror image.
So
We can move it so it looks good. Okay.
well, I really don't care about being in it. I'd rather.
All right.
They fixed it where it got much better.
So Chris says he doesn't care about being in it, so he just wants the slides in it.
Yeah, that's the most important part.
All right. He says he should. Malcolm says you should be in it. Can you move that cup? Better, worse?
So
Just don't look at it. Say that to me.
what if we sat on something?
Yeah. Can we sit it on?
You'll see it
Chalk.
around here for a
Yeah.
little
There we
longer.
go. Is that better?
Feel like a little bit.
Why do we see it reversed though?
That's what I was trying to figure out. But he says he doesn't see it as mirror image. So.
Yeah.
Are you seeing a mirror image or are you seeing the correct
Well,
image?
I go,
Okay,
people
cool.
might go crazy spending money.
All right.
Or they might learn not to.
Well, I think this is gonna be could be the, story.
Okay, we can move it back. We've got it propped up on
Yeah,
something.
they were teetering anyhow. Mailing, yeah. Everyone
Okay.
drop off.
He just stepped out to grab maybe a book or something. Oh, AV card. Perfect. All right, plug it back in too, because it will die.
I mean, that's weird if they have food out there.
Yep.
Yeah.
Yeah, a little bit, but that's it without stretching the cord out.
So the other one's
Stay away. Here's the pole.
Is these communities spread by,
Donuts. They go through hot grease, don't worry.
I don't care.
Okay. Chris, where would it stand? Because
My
you said
last
stay as
thing.
close as possible to
Here's
yourself.
my last thing.
Yeah.
I enjoy it. Here you go, Christine.
All right. All right. All right. Will do. Dr. Shuck says good luck and you look very handsome.
Thank you.
Oh, he can see you. Good.
All right. No problem. Thank
Oh,
you.
that's the haircut.
Oh, we're having a little trouble.
I know, that's why I said it will walk in my.
Did your other meeting get canceled? Huh? Or did you not have did your other meeting get canceled?
Yeah.
Just we're gonna turn the lights down.
Do we need to turn the lights down?
Yeah.
This is a really bad situation. I think we should all start.
How do I turn the lights down?
Ah, That's a good question. There's switches over here to play with. That's okay, they're all turned on. Is that too dark?
Probably for the TV. What does it look like on the?
It looks pretty dark.
Yeah, it's gonna be too dark.
Well, you can see it on it, that's good.
How's that? Good?
Is that looking on there?
I was looking at the screen.
You can look on that screen,
On
John.
the computer.
So Andy just messaged me and said that people have to click on subscribe to the biology department for it to work.
Oh, it's appearing backwards.
Well, we
On
know.
Malcolm said it's fine on his.
Yeah,
I
so
can tell them, they can't hear you.
Okay, so are we ready? Okay, I hope you can see me out there. For those of you in the YouTube world, I'm not sure how to let people know. If you can't see me, you have to click on subscribe to see me. But if you can't see me, that won't do any good. Okay, so it's been a long road getting here. I've known Chris since he was an undergraduate in my genetics lab. And he was such a good student there, I invited him to do an undergraduate research project in my lab, which turned out to be very successful, and he was a co-author on a genetics paper from that. And I think that instilled the bug of fruit fly and genetics into his veins, because he couldn't leave after that. He came to my lab for a master's degree where he studied a mutation, very interesting mutation that disrupts nuclear shape and chromosome segregation. And then after a brief stint as an ecologist with Martin Suey, he came back to my lab to do his PhD, where he's done really an outstanding job defining some of the requirements for meiotic pairing. This has been a really difficult system to work in, and my colleagues around the world have had trouble getting these techniques to work. So Chris was really the first one to successfully get these to work in meiosis, and it's led to some very interesting results. So I'm really sad that this can't be more of a celebratory event in this room, but I'm sure all of his friends and family are out there watching. So without further ado, Chris.
Alrighty. Thank you, Dr. Tom Kildine, for that warm welcome. And I'm very happy to be here today to share with you meiosis is the production of sex cells. Meiosis is two different divisions, appropriately named meiosis 1 and meiosis 2. So before meiosis 1 begins during interphase, all of the DNA in the cell will replicate. All right, and then during prophase 1, all the DNA condenses into a chromosome shape that we're used to seeing. All right, and then homologous chromosomes, all right, they're the same size, shape, and encode the same genes. All right, so during meiosis 1, we have to segregate our homologs away from each other into separate cells. So at the end of meiosis 1, we only have one of our homologs in each cell. All right, and that's called the reductional division, because we're reducing the diploid number of chromosomes to haploid number. Excuse me. Then after meiosis 2, we split our chromosomes that are left. The sister chromatins will separate at the end of meiosis 2, and then we'll have 4 haploid daughter cells that have arisen from 1 diploid parent cells. And it's important to have our sex cells as being haploid. So during that magical moment called fertilization, all right, we restore the diploid number in the zygote. And then that zygote will in turn to start to divide by mitosis. However, I want to point out on this slide the big difference between metaphase 1 and metaphase 2. So we see in metaphase 1, the homologs have to line up across from each other at this metaphase plate, which is just the line of division, the axis of the cell. Take a minute and really appreciate all the mechanisms and events that have to occur for these homologs to line up across from each other at metaphase 1. Cell morphology changing, they have to actually locate their partner, right? Then they have to pair with their partner, right? When they find them, they have to stay with them, right? So a lot of times they'll conjoin, all right? So they have some sort of mechanism for keeping the replicated homologs together. In many species, recombination is involved. Okay, so the homologs will line up next to each other after they've paired, and a proteinaceous structure called the synaptonemal complex will come in and bind those 2 chromosomes and hold them together. And then double-stranded breaks will happen on the sisters of the homologs. They cross over and form these chiasmata, which are physical attachments that help the chromosomes stay together as they move to the metaphase plate and orient correctly so they can separate and go with the opposite cells. All right, then they also have to then disjoin and segregate at the end of anaphase 1. All right, so then at the end of a normal meiosis, you have 1 of each replicated homologs in each cell. All right, and then like we said before, at the end of meiosis 2, each cell will have just 1 copy of the homolog. All right. However, if something happens at meiosis 1, let's say our chromosomes never find each other, or once they find each other, they aren't able to stay together for some reason. Well, that could lead to a meiosis 1 division where both of the replicated homologs go to 1 cell and the other cell gets nothing. All right. So at the end of meiosis 2, once that's occurred, Now we have two daughter cells that have two homologs in it and two daughter cells that have nothing. All right, we refer to these as diplo and these as nolo sperm in the case of male meiosis. All right, so this is a karyotype from a human. All right, so we have 23 pairs of chromosomes as humans. All right, and so in this karyotype, we see that these are the homologs, so the chromosomes are the same size and shape, and again, they code the same genes. All right, but if this segregation occur of chromosome 21, and so the sperm or egg had that extra chromosome, so then when it fertilized, this patient now has 47 chromosomes. All right, and as most people know, somebody with Down syndrome. I had a family friend growing up that I always considered like a cousin to me, who had Down syndrome, and I always kind of attributed being around her and her being in my life is why I was interested in meiosis to begin with. Okay, so that being said, let's switch gears now. Let's talk about the important person in the room, and that's the Drosophila. They have a short life cycle. They're relatively inexpensive, easy to manage. All right, there's also a meiotic advantage. All right, so they only have 4 homologous pairs of chromosomes compared to our 23 and many other high order species. All right, so it's not as much going on in there, right? Not as much chromosomes to follow. All right, and there's also sex-specific advantages. All right, so females, they undergo recombination-dependent meiosis, so they have the synaptic complex. They have the crossing over the chiasmata to help regulate their chromosome movement to the metaphase plate. However, the males do not use any recombination. No synaptic complex, no double-stranded breaks, no chiasmata formed. All right, so they have to have some sort of other way to maintain, right, that gives us some sort of phenotype we can see in the fly. And we get a fly out of a cross, and that fly has that phenotype. We know for sure that that fly got the entire chromosome. So there's no recombination. So we know all the other genes that are present in that fly, which make it really easy to follow particular mutations using markers that are other mutations. And there's tons of different markers in flies, including body color, eye color, wings, bristles, you name it. Okay, so another advantage to studying male meiosis is their progression of spermatogenesis. So this is a testis that I had dissected out of maybe a 2 or 3 day old male fly. All right, down here is what we call the tip of the testis, and it wraps around like a ram's horn. Okay, and then this is the sperm duct. All right, so follow me over here to this picture. So this tip right here of the testis has germ stem cells. Okay, so during spermatogenesis, the sperm stem cell will divide. One of the cells will by mitosis. One of the cells will maintain stem cell fate. The other cell will become what's called a gonioblast, which will then undergo 4 mitotic divisions. All right, and make a cyst with 16 what are called primary spermatocytes at this time. So they're all the same size. They're all encapsulated in this nice cyst. So it's a really nice organized way of looking at meiosis. All right, so then after meiosis 1, the primary spermatocytes will divide into 32 what's called secondary spermatocytes, also still in that cyst. And then again, at the end of meiosis 2, we'll have 64 what are now called spermatids, which eventually differentiate into to sperm. So it's really nice if you're interested in looking at these primary spermatocytes, secondary spermatocytes, you can simply dissect the male and use a needle to cut the tip of the testis off, and all of your cells in those stages will pour out onto the slide, and you can fix them and stain them and do everything that you want to do. It takes a little bit of practice dissecting fruit flies, but after a while you get to be good at it, after 10 years. Okay, so another interesting aspect, so you might imagine during meiosis in Drosophila, if we don't have those landmarks of the chiasmata and the synaptic complex, right, we have to study a different way to stage the progression of prophase. So we use an author named Giovanni Cinci, who in 1994 went through and named the stages of progression of prophase. All right, so S1 right here, that's that primary cyst, right? So after what I showed you, right after the 4 mitotic divisions, our primary spermatocytes. Okay, so you see as they grow and get bigger through S2A, this is the chromosomes, all right, of one cell here. As we move from S1 to S3 and S4, they look kind of like domains, right? So all like materials being sequestered in specific areas of the cell. Well, it turns out that each one of these domains is one of the major chromosome pairs. All right, so this could be chromosome 2, this could be chromosome 3, and this could be the X and Y bivalent. And the chromosome 4 is very small. It's also autosome. They usually kind of associate with the X and the Y. All right, but it's just really interesting to look at what's going on here. Like, why is this even occurring? Why is the DNA being sequestered into certain parts of the cell during prophase? Okay, and we'll come back to that a little bit later. Okay, so now let's look at Drosophila recombination independent meiosis. So what's known now before the research that we've conducted in the lab? Well, we know that again that there's 3 pairs of autosomes and 1 pair of sex chromosomes. So here labeled the thicker gray bars are heterochromatin and the thinner black bars are euchromatin. Right, so we see that the autosomes have both heterochromatin and euchromatic homology, whereas the sex chromosomes only have heterochromatic homology, so there is no euchromatin on the Y chromosome. All right, but other than that, the males also developed 2 genetically separable pathways to segregate these chromosomes, and both cis and trans factors vary between these. So I want to take a couple minutes to go over this. Right, but at this point, I really want to stress to you, because I'm going to be using the words pairing and conjunction quite frequently, really understand what they mean so you can follow what we're doing. All right, so pairing, again, is the initial interactions required for homolog recognitions and association. So you're in a room, hey, there's my partner, right? And then this is how they come together, not very well understood in many different systems. All right, but again, once they pair, they have to conjoin. Again, we talked about in recombination, we had the chiasmata that help. All right, but again, in Drosophila, we don't know exactly how the system works. All right, but conjunction is the ability of the paired homologs to remain together. So whatever system that they have. All right, so what's known? So when it comes to autosomal pairing, right, the cis-actin components, all right, heterochromatic homology is not associated at metaphase 1. So a lot of these studies, I failed to mention this, a lot of these studies are done after early prophase. So pairing begins in early prophase. All right, the S1, S2A stage, right? But because of technological limitations, a lot of times it's been hard to access cells at that stage. All right, so this is a study done in 1979 that used these are just free arm chromosomes here that have heterochromatic homology and euchromatic homology. All right, and we see that they're associated here. All right, and there's no homology between this compound chromosome 2, and you see it's over here in the cell by itself. All right. But then, however, when we have both euchromatic and heterochromatic homology here, and then heterochromatic homology on this chromosome 2, we still see that they are not paired at metaphase 1. Okay, insinuating that heterochromatic homology is not associated at metaphase 1. All right, so you might imagine if they're not together at metaphase 1, they're not able to segregate. So heterochromatic homology alone is not able to segregate our autosomal homologs. So the only study to date that really looked at pairing and that prophase 1 in early when pairing actually begins was a study done by Vasquez in 2002, where he uses the LAC-O, LAC-I system to help him study pairing. So there's these LAC-O tandem repeats. Okay, this is borrowed from bacteria on the chromosome 2 and 3. So each one of these represents a different chromosome and a different array of LAC-O sites. And then what you can do is you can express the LAC-I transcription factor attached to a GFP protein that allows green fluorescence in the cell. And if you have a transgene and activate that during meiosis, you can actually see that the LAC-I will bring the GFP to the LAC-O, all right, and light it up. So you can see here, like in this area, this chromosome is paired because we only see one signal. So that shows the chromosomes are on top of each other. Whereas here we see 2 signals. All right, and again, he used 3 different, now these are exogenous sequences, right? These are natural to the fly, right? But what he found by mid-prophase 1 that the pairing was lost, which kind of shows that, okay, well, they were pairing at these euchromatic sites, but then the pairing is lost after the middle of prophase. So it kind of leads you to think that, well, maybe there's something else that's maintaining them at that point. Okay, and again, that's the only study to date that's actually been able to look at pairing at early prophase when it actually begins. All right, so let's talk about sex chromosome pairing for a minute. All right, like we said, sex chromosomes don't have any euchromatic homology. All right, so they can't pair there if it's not there. All right. However, originally regions near the centromere were shown to be associated at prometaphase 1. So some research showed that here in this wild-type cell, and this was a study done in 1990, we see our X and Y attached here. Well, this chromosome here, this X chromosome was missing the rDNA sequences. And when the rDNA sequences were missing, we saw that the X and Y were no longer paired. And this is now, again, at late prophase. Okay, so not early prophase, at late prophase. All right, and further studies using transgenes identified that it's actually this 240 base spacer region between the transcription units of the rDNA that is required for the pairing. All right, so because whenever those goes away, they're no longer able to segregate and find each other. So it's kind of interesting that there's all this heterochromatic homology, but it's just these individual sequences, these IGA sequences that are required for sex chromosome pairing. All right, so besides those those cis-acting components, there's also some trans-acting components, including 3 proteins to date have been identified to be involved in meiosis: MNM, SNM, and Teflon. All right, so interestingly, autosomes require all 3 of these chromosomes to have proper all 3 of these proteins to have proper chromosome segregation, whereas the sex chromosomes only require MNM and SNM. Right, so there's already some sort of difference between segregating autosomes and sex chromosomes within the same species sex. Right, it's very, very interesting. And MNM has been shown to bind euchromatin along the arms of the autosomes. Okay, very similar, right, to what we were talking about just a minute ago, where they were paired. All right, and SNM and Teflon have never been identified, not able to show them attaching to chromosomes, but that could be because of technical limitations. They might actually be binding those. All right, MNM and SNM mutants do not affect pairing, only segregation. Okay, so that's interesting. So it's like these proteins are needed, but they're only needed at a certain point. All right. So finally, it's like, well, maybe the rDNA euchromatic sites along the autosomes are conjunction sites in addition to parent sites, right? So remember, if you conjoin with something, right? If you're conjoined, you obviously had to pair to begin with, right? So maybe what we've been talking about, these pairing sites that have been identified in the later prophase, might actually be not only pairing sites, but also conjunction sites for these conjunction proteins, which exist basically to form the same kind of function that the synaptic-emal complex does in recombination proficient meiosis. Okay, so let's just go over real quickly, a little summary. All right, so in order for homolog chromosomes to pair, they first have to find their partner in a pair, and then conjoin, and then orientate, and then segregate. All right, all those steps are required. So what we know right now is that euchromatic sequences are important for the autosomes. All right, MNM, SNF, and Teflon are required for segregation of the chromosomes. MNM's been shown to actually bind these sequences along the euchromatic arms of the autosomes. All right, and then the sex chromosomes, something special about the rDNA, right? So we know that that's required for segregation. And also we know that MNM and SNM are required, and they also have been shown to bind the rDNA. Okay, so after all this being said, my question is, the whole direction of my project is, are there other sequences that can pair at early prophase 1? All right, so we know that on the sex chromosomes, the rDNA can pair, all right, and we know on the autosomes, euchromatic homologies can pair, all right, but maybe those were just conjunction sites that were identified later. Maybe there are other sequences that can also pair, all right? So my aim 1 is, can euchromatic homology facilitate XY pairing, conjunction, and segregation? So I want to separate all of these steps. All right, and now I want to make euchromatic homology exist between the X and the Y. Say, okay, well, if it was there, would it be able to pair? Okay, and all of this information and the images, especially if you can't see them there at home, were published in Genetics this month. All right, so again, we have to have a euchromatin-mediated XY pairing system. All right, we have to make one of these, right? All right, so originally this is how X chromosome pairing occurs. So this is a wild type X, all right? So this is heterochromatin in blue. We see our important rDNA, and euchromatin in red. And then a wild type Y, which again, it's all heterochromatin, and there's our rDNA. So there's usually where the X and Y come together, all right? And those conjunction proteins bind. All right, but also this chromosome exists called the rDNA deficient inversion 1 SCUT4L SCUT8R chromosome. All right, and just for purposes of the fact that I have to repeat that about 70 times today, I'm just going to call it the rDNA deficient X. All right, so the important part of it is to say, well, hey, rDNA is missing, right? It's not there. Euchromatin is also inverted based on the way that the chromosome was made. But again, the important part is that the rDNA is not there, okay? And we've seen in other studies that when the rDNA is not present, the chromosomes can't segregate. All right, but now we need a Y chromosome that has euchromatic homology on it. Right, euchromatic homology that matches the X. Well, There's this collection of chromosomes called duplication 1Y BSCs. The BSC just stands for Bloomington Stock Center, where they were made. All right, and each one of these duplication 1Ys, the duplication 1Y name means duplication of chromosome 1 material on Y. Chromosome 1 is the X. All right, so X material is duplicated on this Y chromosome. All right, so this U chromatin right here is duplicated from right here on the X. Okay, so we can actually have a male with these two chromosomes and then monitor his offspring or monitor his sperm, all right, and say, okay, were these chromosomes able to pair and segregate? But there's not just one, we have a nice assortment of all kinds of different duplication 1Y. So each one of these BSC numbers represents a duplication 1Y that has that area of euchromatic homology duplicated on it. Okay, so you can see these are kind of drawn to scale. They have different sizes, all right, and then also in different locations along the arm of the X. Okay, and that'll be important in a minute. All right, so we start off, now we've got our system. All right, we know what we're going to do. First off, we want to see what happens to segregation. All right, so we're going to do genetic crosses. All right, so we're going to have a father fly, that has an RDNA deficient X, all right, and one of our duplication 1Ys. All right, and then a mother bearing genetic markers that's able for us to follow the chromosomes that the sperm like we had talked about earlier. All right, so the male, right, can produce 4 different types of sperm. He can produce a normal X and normal Y if pairing and conjunction segregation happens correctly, right? Or it can, if they never pair or if they can't segregate correctly, get stuck together, something like that happens to homologs. The X and Y will go to the same pole and the other pole will get nothing. Okay, so these are the 4 different classes of sperm. And these are just down here for you to look at. These, whenever these come out, we can actually look at those markers that we were talking about before. And I can say, okay, if this male has bar red eyes, I know for sure it has a duplication 1Y. Okay? Right. Unfortunately, though, we have to eliminate 2 of these sperm classes right off the bat because the duplication 1Y is aneuploid, right? It's got extra duplicated material on it. All right. Whenever you're aneuploid, a lot of times the gene dosage imbalances occur, which causes a lot of the phenotypes we see in human aneuploidy as well. All right, so that causes viability difference of these offspring. So I didn't want to count these offspring if they weren't all possibly there. All right, so instead we had to come up with this other formula where we just used the nullos over the total of the X and nullos to get our segregation rate. Okay, so let's take a step back and look at just a bunch of numbers. So remember, if the X and Y don't come together ever, or if randomly, 25% of the time they'll go the other way, 25% of the time they'll go to one pole, 25% of the time they'll go to the other pole. Okay, so completely random segregation has a frequency of 0.5. Okay, so all of these numbers are lower than 0.5, right? So that shows that the segregation is not completely random. So something's going on here. But the major difference I want to show you is look at the difference between this guy and this guy. Right, this guy barely has any segregation issues at all, only 8% non-disjunction. So even though the rDNA is not present, the euchromatic homology is making these, the RDNA deficient X and the duplication 1Y segregate from each other. All right, so we want to look, all right, well, what's special about different between these guys? Well, we talked about earlier, they were different links. However, when we plotted these out, this non-disjunction frequency versus the amount of euchromatin present, no relationship. All right, so it wasn't like more euchromatic homology made you segregate better. Okay. However, when we plot these based on where the location of the homology was duplicated from on the X, we see this interesting relationship where the closer you are to the centromere, the better you are to segregate, capable to segregate. So the farther you go away, not able to segregate quite as well. All right, so that's very interesting. And we'll come back and talk about that in a few minutes. All right, so there are some caveats to these genetic tests that we did. I've already talked about the one, right? That duplication 1Y viability differences. So because the sperm is aneuploid, it can affect the viability of the zygote that gets that particular chromosome. All right, but another one is this mysterious process called meiotic drive. Right, it's not very well understood, but basically what it is that sperm with lower chromatin content are preferentially recovered over sperm with higher chromosome content. Okay, so that might mean, all right, well, if we're looking at outcome, right, we're looking at sperm based on which ones were fertilized eggs that could grow and live. All right, if some classes are being preferentially recovered, that's going to adjust my segregation rate. All right, so I wanted to know the actual true non-disjunction value. All right, so I was like, well, how could we do this? Well, we decided to look at it cytologically. All right, so we're going to use fluorescent and C2 hybridization. So from here on out today, I'll just refer to that as fish. All right, but we didn't just do any kind of fish. We wanted to have these new high-end kind of probes, like they're single copy probes. Okay, so what this is the double helix of the DNA. All right, and what we can do is we pick a million base pairs that we're interested in probing. All right, and we send off that sequence to a company called Arbor Bioscientists. It was formerly Microarray. All right, and they actually scan the genome and they make probes that are 40 to 50 base pairs long, right? About every 1,000 base pair in that million base pair span that we provided them. But they assure that those probes won't bind anywhere else in the genome. Right, so they spend, that's where all the money comes in. They spend all this time actually searching and making sure that they won't bind anywhere except where we want them to. All right, and at the end of each one of these probes that are 40 to 50 base pairs, is this triply labeled floor. All right, and what happens is, and then we also have so that's for the X probe, so that's going to label our X chromosome, and then for the Y probe, again, it doesn't have any euchromatic molecule. We just use the repeat probe to the AATAC segment, which is repeated on the Y chromosome. All right, so what happens when you dissect flies? Right, you find the cells that you're interested in, and you undergo this FISH protocol, which only takes about 36 hours to do, so not very long. You get these pretty beautiful images, not as beautiful at home, but beautiful images where we can actually follow what's going on with the X and the Y. Okay, so in this case, what we're looking at here is a meiosis 1 division, right? Our X is in red, our green is the Y, and the blue is the DAPI. It's the DAPI standing for all the DNA. All right, and so what we can do is, just going to show this again to remind you. So this right here is a dividing cells after meiosis 1. Right, so this would be kind of late telophase. You see the cells are far apart. All right, here in this panel, we see normal disjunction. All right, so this got the Y homolog, and this cell got the X homolog. Right, this is meiosis 1. Remember, homolog separated. However, here we can see, well, what happened here? Well, something happened. The X and the Y didn't segregate properly. So either they never paired, or they weren't able to conjoin, or something happened. And this cell got both the X and the Y, and this cell got nothing. And you can actually see a size difference in those two if you can see it from there. All right. So then once that happens, all right, we can score all the cells that we want to in that stage. All right. And a lot of times they're encapsulated in those cysts if you dissect well. So you'll have 32 of those cells right next to each other to score. All right. And then also we can score non-disjunction at meiosis 2. So here is what happens when a normal meiosis 1 occurs and meiosis 2 goes in. So here this is our separating our sisters of the X, only in one cell at the end of meiosis 1. This is making your diplosperm, and this is making your nolosperm. And this was captured just right at the beginning of anaphase. So you see this cell here, this nolo, is just now splitting, and these are a little bit farther ahead. So if you're wondering what the difference between those are. Okay, so now I can actually go in and count what's going on in meiosis instead of having to look at just the outcome. All right, and what we found is that cytological analysis confirms the genetic analysis of the non-disjunction. Okay, so we still see that same relationship between the three duplication 1Ys that we see over here. All right, also failed to mention that we were only able to get probes for certain chromosomes. We couldn't do it for all the duplications, unfortunately. But the important part is the same relationship, right? So this is this data that I showed you before. And this is close to the centromere, and this is the cytological non-disjunction data. So we see that we were kind of overestimating non-disjunction. Notice that the scale is different. But again, it was a nice enough analysis, close enough to show us what we needed to know. All right, but again, we still see that important relationship between the closer the homology lies to the X and its ability to segregate. And again, we'll come back to that. Okay? So, can euchromatic homology facilitate XY segregation? Yes, it can. And now we have cytological analysis that we can do to get rid of some of the issues of viability and myeloid drive. So now that we know that they can segregate, I know we're kind of going backwards, but next I wanted to look at these conjunction proteins that we talked about, right? So, are these conjunction proteins required for my XY euchromatin-mediated XY segregation? And so, if you remember, S&M was required for both the sex chromosomes and autosomes, and Teflon was only required for the autosomes. Okay, but we don't know for sure, is it the autosomes or is it Teflon's required for euchromatic pairing? in segregation. Remember here, sex chromosomes pair through the heterochromosome. So if Teflon is euchromatic specific, we would expect Teflon to have an effect on our sex chromosome mediated segregation using the euchromatin. And if Teflon is just special for some reason to the autosomes, we'd expect no effect of knocking out the Teflon protein. Okay, so on the left here, this is a skewed 4, skewed 8, the RDNA deficient X, all right, and a yellow plus Y, which just means a control Y, all right, so it doesn't have those big region of the euchromatin duplicated on it. All right, so this is in a normal fly. We see that we have high levels of sex chromosome non-disjunction, and again, that's because the RDNA is missing on the X, and we haven't put any euchromatic homology to help segregate. All right, and there is no effect of 4th chromosome or autosomal non-disjunction. All right, and then whenever we have a Teflon mutant now, so now this fly can no longer produce the Teflon protein, we still see that sex chromosome non-disjunction, which really has nothing to do with Teflon. All right, but we now see that the autosomal non-disjunction has risen to almost completely random. And so that's just a test. That's to show us, okay, well, our Teflon protein is definitely working, or not working, I should say, in the mutant. Okay, so now when we use our duplication 1Y BSC76, which is that one that showed the best ability to segregate, we see that, as we imagined, that the sex chromosome non-disjunction is lowered, and that's because of the euchromatic homology, and again, no effect on the autosome. However, now we have a Teflon mutant, right? Again, no effect on the sex chromosome. All right, so that's showing us that the Teflon is not required for our sex chromosome mediated system using euchromatin, right? So that shows us that there's something special about Teflon and the autosomes, not Teflon and the euchromatin. All right, so next we wanted to look at S&M, all right? So S&M basically has already been shown to interrupt the pairing of the rDNA, right? So we already know that, so we're actually able to use a wild type X in this situation. And so this is our control, right? So there's no effect of S&M in the flies that have good S&M, obviously. But then when we mutate S&M, we see the high non-disjunction of the sex chromosome, and then the 4th chromosome non-disjunction is about 25%, which is what's reported in the literature for S&M. So we know that the S&M mutation is working. All right. So if S&M was required for our XY mediated pairing system, all right, then we would expect to see the same result whenever we have the duplication 1Y and an X, and that's what we see. Right, so whatever is occurring between the euchromatin pairing system does not occur, or it does not occur when S&M is not there. So S&M is needed for that as well. Okay, so our conjunction proteins, S&M and Teflon, are required for XY segregation. S&M is needed, Teflon is not needed, suggesting Teflon is autosome specific, not euchromatic specific. All right, and finally, what you've all been waiting for, we're going to actually use our high-quality probes to look at pairing at euchromatic sites between the X and the Y in early prophase. All right, again, this has never been done before. This is using actual Drosophila sequences, not just sequences, lactose sequences from bacteria. Okay, so we have probes. So we got to kind of change our mind thinking this time, right? So the sex chromosome probe is going to be binding both the X and the duplication 1Y. Okay, again, so the homology that we've duplicated from the X to the Y, that probe is going to recognize that, right? So our X and our Y are both going to be probed red, all right? So we can see if they're together, we only see one dot, that means that they're paired, and if the X and Y are separated, there'll be 2 dots, okay? And we're wanting to look at these early early S1 to early S2 cells. All right, and we're going to judge those cells by DNA morphology and also a nuclear area between 10 and 20 microns. Just kind of want to make sure that, there's also some kind of asynchrony between these stages, right? This is just stages that someone made, so it's not always going to be, they're not going to always both separate at exactly the same time. All right, so we want all the different chromosome pairs are not going to separate exactly the same time. So what I wanted to do is we have to have an autosomal probe as a control to make sure that we're actually selecting cells that are still in the right stage. We want to make sure that nobody's progressed on to unpairing of chromosomes. Okay, so this is an example of what we see. All right, so in panel A, we see our X and our duplication 1Y are paired. All right, so we see both signals together, and our autosomal control probe are paired, so that's again on chromosome 2. right? And panel B is an example where the X and the Y are not paired, right? So we still see our autosomal probe is paired, so we're at the right stage, but for some reason the X and the duplication Y are not paired in that stage. All right, and there's other things that we can see, like where the cell probably would not have been scored because you see both the X and Y unpaired and the autosomal probes unpaired. And then E is a rare cell we don't see very often, where this is we attribute this to being one sister that's not paired along with the other three. Okay, and again, we tested 3 of the duplication Ys with their ability to pair with the RDNA deficient X, and we found that they're all able to pair pretty efficiently. All right, which kind of makes sense, right? Because they were able to segregate, right? And obviously they're probably paired. But this was really, really exciting, right? It's the first time that we showed other sites in addition to the RDNA can also pair in early profix. All right, so we wanted to take it one step further and say, okay, well, just because these sequences can pair, can they actually direct segregation? All right, so based on the fact that what we said earlier, whenever chromosomes don't or segregate randomly, 50% of the time they'll go to the correct poles and 50% of the time they'll go to the wrong poles. We can use that value along with cytological non-disjunction that we've scored ourselves to come up with the percentage of the paired cells that actually disjoined. All right, and what we found was, all right, so these are the pairing frequencies. These are cytological non-disjunction frequencies that I calculated looking at cytologically. All right, we found that the paired sequences can direct segregation. All right, and the interesting thing was something reappeared again. Well, the ones that were these duplication 1Y sequences that were closer to the centromere from the X were actually able to pair perfectly, or pair and segregate perfectly. All right, and then as we get farther away from the centromere, we see it's not as able to segregate as well. And this one over here is even less able to segregate, even though they paired to begin with at a higher frequency. So what we think is going on is that pairings closer to the centromere are better able to satisfy what's called the metaphase checkpoint, right? So if you're thinking about during metaphase, you got the 2 poles on each side, all right? So you want to segregate one homolog to this pole and one homolog to this pole. Microtubules come out from those 2 poles, right? And one from this pole will attach to one homolog, and this will attach to the other homolog, all right? So if they're paired closer to the centromere, the homologs, all right, it's going to make more tension. It's going to create more tension between those 2 attachments of the microtubule, which kind of sends a checkpoint to the cell saying, hey, everybody's where they're supposed to be, all right? But if you have pairings farther away from the centromere, way down here, it's harder to feel that tension. So that might slow down the cell cycle a little bit. They may never be repaired, and eventually the cell cycle will go on without them actually ever being resolved. Okay, so that could explain the differences we see between the abilities to segregate based on where the homology of the pairing sites lie. Right, so again, that's just our speculation. All right, so the last thing that we wanted to look on this aim is this: can we define the minimal requirements for pairing and segregation? Okay, so this is our duplication of 1Y BSC 76. So again, the one that paired and segregated the best, or had segregation frequency of only, non-disjunction frequency of only 8%. All right, it's 1,500 kilobases. All right, so I looked and I found, well, there's actually a couple of other duplication 1Ys that exist that are smaller and actually nested in that same euchromatic homology that was in BSC 76. All right, one of them is 120 kilobases, the other one is 161 kilobases. So I also wanted to test these ability to pair and segregate. All right, so the pairing abilities we found were actually higher than the longer sequence. Right, that could be attributed to what a couple of authors have talked about in the literature is there could be anti-pairing sites that also exist. So this sequence here is able to pair, but maybe not pair as well because there could be other factors interfering with the pairing. All right, but again, the important take-home part is as short as 120 kilobases, it's still able to pair. All right, and then when we do our calculation of direct segregation, we also find that all 3 of them are able to pair and segregate. Okay, so to sum up, aim 1, all right, euchromatic homology can facilitate XY pairing. So again, we gave the X and Y bivalent pair euchromatic homology that's not normally there. It was able to pair. Pairings closer to the centromere are better capable of directing segregation. We know that the minimal homology required for pairing is less than 120 kilobases since it's still paired. Our internal fragment that I just showed you is still paired. Anti-pairing factors may exist. Euchromatic homology can also direct segregation. All right, so it's not just pairing, it can also pair and segregate. And we know that SNM is required, and we know that Teflon is not required. So now we have this great tool, right, that we can use to study pairing in male meiosis when pairing is actually established. We have tools that we can use to get rid of confining aspects of genetic testing. All right. So I started thinking a little bit more about, this is a cell here, this is showing the domain formation. This is a picture of the domain formation. So remember what I showed you at the beginning when those bivlets kind of moved into separate sections. All right. So I was thinking, well, we put a chunk of the X on the Y and it was able to pair and segregate. But I wonder what would happen if we put a piece of X on one of the autosomes. All right. So we've already showed you that those go into separate domains. I wondered if, well, if we have homology between what are called heterologs, all right, will we still be able to see pairing, conjunction, and segregation? All right. We know a little bit about that from a study done in the early 90s, all right, that looked at what duplication 2Y chromosome. So that's chromosome where some of the autosome material from chromosome 2 is duplicated onto the Y chromosome. All right, and this author studied it in mid-metaphase, so again, after the initial pairing had begun. All right, and what he did was he measured the frequency of these quadrivalents. All right, so quadrivalent meaning that the X paired with the duplication 2Y at the rDNA that's present. The duplication 2Y paired with chromosome 2 at the material that was duplicated on the Y. And then the intact chromosome 2 will pair with a deficiency 2, which is just the part of the chromosome that was duplicated over here. And he would count how many times all 4 of those were associated in the cell and found that they weren't there quite frequently. So they were able to pair. So this duplication material from chromosome 2 was able to onto the Y and made the Y pair with the 2. However, and this is just showing he tested a bunch of different segments, kind of similar to what we did. All right, and he found that all the segments that had euchromatic homology was able to pair, but the one fragment that didn't have any heterochromatic or only had heterochromatic homology was not able to pair, which is what we kind of already know from the autosomes. But the interesting thing was when he measured the segregation, it wasn't very much higher than random. Okay, so able to segregate a little bit, but not very much. Okay, so that made me start to think, well, if they're paired, why are they not segregating? All right, so that brought me to my next aim, all right, which is, are the pairings between heterologs resolved during domain formation of prophase 1? Okay, so this domain formation that we were talking about before, right, we start to see at this stage, we start to see these domains. Well, maybe that's Drosophila male's way of separating all those heterologous pairings that are incorrect while still maintaining our pairings between our homologs. Okay? So the first question was, can euchromatic homology direct X duplication 1,3 segregation? Right, so you might be saying, well, why did you choose duplication 1,3 and X when there were some other chromosomes that existed? Well, unfortunately, the duplication 2Y chromosomes no longer exist. All right, and in my case, I also wanted to look at XU chromosomes, since that's what we had studied before. And also, these duplication 1,3, so as you might imagine, they were just duplicated X material on chromosome 3, are really a nice system because they were all put in into the same site in the chromosome 3. So each one of the duplication 1,3s has its X homology in the same exact location in the 3rd chromosome. All right, and also the sequences have been confirmed and we know for sure exactly what's there. All right, so again, we're going to take a piece of the X and duplicate it onto chromosome 3. All right, so this is DC398. All right, and these are the other duplication 1,3s that I tested. And again, this is kind of trying to scale how long the euchromatic homology was, and then also in relationship to the centromere. And then these ones with asterisks are the ones that we were able to study using the pairing assay. All right, but first let's talk about segregation. So I did all of these tests. We had to do female controls. Did all this work, found out, well, the duplication 1,3 and the X, they don't segregate very well. Some of them are a little higher than 50% random, but the highest is only 60%. Remember we saw segregation of duplication 1 and Y, and X, some of those were as high as 92% pairing and segregating. So only 8% non-disjunction. All right, so again, this is kind of similar to what was seen with the duplication Ys, right? That they were not able to segregate, but then not able to maintain that pairing all the way through to segregation. Okay, so we did our pairing test again. So this is again the S1A cells, all right, and we're using our sex and duplication 1 probe. It's going to be red, all right, so it's going to recognize the sequences that are duplicated on chromosome 3. And so we can study the X pairing with duplication 3. And again, we have our control probe for the autosome, again, just to make sure that we're staging the cells correctly. All right, and what we found was, well, all the duplication 1,3s that we tested were able to pair with the X at very high frequencies. Okay? So, is euchromatic homology between heterologs capable of pairing? Yes, it is. It definitely is. But for some reason, it's not able to maintain that pairing to segregation. Right? And we didn't see any relationship between pairing and centromere proximity, which might make sense because they didn't remain paired to segregation. Okay? So finally, big question, is pairing between heterologs lost during chromosome domain formation? So is it because of those chromosomes moving into their separate sections of the cell? Is that what helps regulate the differences, or the pairing between heterologous chromosomes? Okay, so again, this is our domain formation through prophase 1. Earlier, we were studying cells in S1 to S2A, so right when pairing is first established. But now I want to look at cells in the S2B stage, so right when those domains are first starting to appear. Okay, so what we're going to do is, in the study, we're only able to look at cells where the green signals are unpaired and the red signal is paired. So the reason why we're doing that is if this is during S1, all right, we have the X and duplication 1 paired and our chromosome 2 control probes are paired. All right, again, they might not always separate at the exact same time. So there could be some times when the red unpairs before the green and sometimes when the green unpairs before the red. All right, but in this case, I didn't want to score any cells where the reds hadn't paired to begin with. Okay, so in this case, right here, these reds may never have paired to begin with at S1. So I don't want to say, well, they were separated at S2B if they were never paired at S1 to begin with. Okay, so we're only able to score cells where the red signal was paired and the green signal was unpaired, saying that the autosomal pairs have progressed on into the S2B stage. They're starting to become unpaired. All right, while the duplication 1,3 or duplication 1Y are still paired. All right, so we're going to compare. Does the duplication 1Y remain paired with the X longer, or does the duplication 1,3 remain paired with the X longer? Okay, so on the left here, we have the frequencies of cells in which the autosome has unpaired and the duplication remains paired. Okay, so that's kind of a control with S1 to S2A. We look, now again, remember, cells are supposed to be paired in S1 to S2A, but occasionally the cells may have progressed a little bit earlier and the autosomal probes may have come unpaired earlier. All right, but what we find is that these are comparing two to the different duplication 1,3s, comparing two to the duplication 1Ys, but even in S1 to S2A, when the autosomal chromosome paired is unpaired, there are more duplication 1Ys still paired with the X. Okay, so saying in other words, duplication 1Y is pairing with the X, duplication 1Y is pairing with the X, all right, and we want to know which one of those comes apart first. Sorry. All right, so then when we look at S2B, the most important stage, all right, so again, all of these autosomal probes have come unpaired, but we're looking at the frequency at which duplication 1,3 is still paired with the X, and the duplication 1Y is still paired with the X. We see there's no difference between our two duplications 1Ys and no difference between our two duplication 1,3s. What we do see is a big significant difference between the duplication 1,3s and the duplication 1Ys. All right, which again, these are just snapshots in time. All right, this would be great to do in real time. These are just snapshots in time, but out of the snapshots that I've captured, when the green autosomal control probe is unpaired, more of the duplication 1,3s are unpaired than the duplication 1Ys. Okay, so that suggests that possibly there's some mechanism that separates those heterologous pairings, but I like to suggest that it's because of the domain formation. So the domain, as its chromosomes are moving apart in the cell, heterologous pairings come apart while the correct homologous pairings stay. Okay. So our aim 2 summary, euchromatic homology between the X and chromosome 3 can pair. There's no relationship between pairing and segregation. All right, euchromatic homology is not very effective at directing segregation. All right, we found more cells with duplication 1,3s were unpaired from the X at S2B than were duplication 1Ys at S2B. And again, we suggest that pairing between heterologs is most likely lost during chromosome domain formation. Okay, so it would also be really nice to be able to study that in real time to be able to tell for sure if that's what's going on. All right. So the last thing, we can use these new tools. All right. So we can now look at pairing in early prophase 1 between homology sequences. We can look at the creation of sperm inside, right, without having to look at the outcome of the sperm being able to fertilize an egg. All right. So again, the genetic tests are time-consuming, laborious, and indirect, right? So like, just as an example, when you study a mutation like MNM and SNM that causes random segregation of all the chromosomes, you have to do a lot of crosses to get enough progeny to survive to make the values substantial or significant. All right, massive non-disjunction can lead to sterility. Okay, so can we use this fish pairing assay to directly assess uncharacterized meiotic mutants? Okay, so a collection of all kinds of different meiotic mutants exists. All right, and a lot of times you'll take one of those mutations in a fly and you just start doing segregation assays and try to see what happens. All right, so I was thinking, well, what if we took one of these mutants and just did the fish pairing assay on it to see whether or not we could see, maybe figure out what's going on. Okay, so I'm going to show you guys a slide. I'm not keeping up with my time, but we didn't start on time. So I want to show you guys a slide. I'm going to take 10 or 15 seconds to look at it and see if you can tell something, anything that's going on. And this is, again, this is going to be a secondary spermatocyte cyst, so after meiosis 1 division. Right, our X chromosome is going to be marked red, and our Y chromosome is going to be marked green. So everybody take about 15 to 20 seconds, look at this image, and see if you notice anything unique. So Dr. Leprey, since you have not read my dissertation, do you notice anything unique about these
They're not all
cells?
the same.
Well, they're not all the same, right? But which cells do you see more frequently than the others?
Well, the most frequent ones, of course, are the ones that are out there.
What, so you're saying with the red and the green signal?
Yeah.
Right, so what's weird about that? This is at the end of meiosis 1. At the end of meiosis 1, you're supposed to segregate homologs away from each other, right? So we would expect to see cells that are green and individually. It's just like, what in the world's going on here? All right, so it's like sister chromatids are supposed to segregate with each other during meiosis 1. The homolog phenotype that's never been seen before in flies at all. And in yeast, it's similar to what's called the monopolar complex, right? Which is a proteinaceous structure that helps maintain sisters during all the meiosis chaos to make sure that they segregate together at meiosis 1. Right? And any mutation in any of the proteins in that monopolar complex will cause sisters to segregate the other way. Right? The gene that's of this mutation is known, and this gene is not it's not found in yeast. So maybe this is the fly's way of making sure that their sister chromatids separate at the correct division, meiosis 2, and not meiosis 1. So this is a type of aspect, right, we would have never been able to see from genetic testing. I mean, we had a little bit of genetic data on this that showed basically completely random segregation. We would have never in a million years been able to guess that's what was going on there. Right, so this is a really nice tool. And again, I told you, it only takes like 36 hours. You do the dissections, you stain it overnight, you wash it, and you spend the next 2 or 3 days counting cells on the cans. Right, Lee? Right. Okay. So again, it's really, really, really exciting. So that's what the lab has to look forward to. Additionally, besides me leaving. All right. So let's go ahead and talk about the conclusions of my project. Okay. So for the first time, we showed that any homology appears to be able to facilitate pairing. Requirements to maintain chromosome conjunction remain undefined, but we know that in this X system, XY euchromatic pairing system, the SNM is required, F1 is not. More research obviously needs to be done to determine other factors for pairing. All right, a mechanism for disrupting pairings between heterologs definitely exists, and it may be related to the domain formation that occurs throughout prophase 1. And finally, that being said, I'd like to thank the lab members that actually helped me contribute to this work. Former lab members Katie Hansen and Andrew Bourgeois, and current lab members Sarah Pellizzari and Sean Thornton, who did the work with the monopolar mutant I just showed you. Kevin Cook at the Drosophila Stock Center for making these very, very useful duplication 1Y chromosomes. Obviously, my family and friends, my mom and dad, Christy, Andrea, and the other lab members that have helped me. My dissertation committee, Dr. Catula, Dr. Stanley, and Dr. Lajeunesse, who I hope is feeling better. wonderful lab advisor and committee chair, Dr. John Tomfield-Dean, who I definitely 100% could say would not be standing here without him. And then the funding from UNCG, and then also Dr. Tomfield-Dean's NIH grant. And with that, I'll take questions.
Talk about whether the euchromatic sequences that you put on the Y chromosome, are they equivalent really functionally to the ribosomal sequences in terms of what's going on with the pairing between the X and the Y?
Functionally equivalent.
Yeah, equivalent. That
So what I think is happening,
meaning you made it's artificial, really. You put on that euchromatic. But is it functioning in the same capacity? Is there anything different about having the ribosomal versus, that is, do the chromosomes behave differently?
so
Could you repeat
Yeah,
the question for the online audience?
right, so Dr. Catula was actually asking me if there's any functional difference between the euchromatic material that's duplicated on the Y and then the endogenous rDNA pairing sites that are on the Y. Well, so I think the main reason why the euchromatic material that's been translocated, all of them showed the ability to segregate at least at some point, and all of them that we tested showed the ability to pair. But remember, the Y chromosome doesn't normally have that euchromatic homology. So the rDNA is the only site where homology pairs. So I actually did do another test I didn't have enough time to talk about today, where we had a normal X chromosome, right, that has the rDNA and then the euchromatic homology. And then we used one of the duplication 1Ys that has the euchromatic homology on it. And we showed that whenever they're able to pair both the rDNA and the euchromatin, it didn't have any other effect. So meaning that it was still able, like the euchromatic homology doesn't decrease rDNA pairing, or rDNA being present doesn't decrease the euchromatic pairing. So I'm thinking that the rDNA kind of evolved as the natural pairing and conjunction site because the rDNA is located closer to the chromosome. And there is no other, euchromatic homology on the Y.
Yeah,
So
I
the
mean,
pairing,
that's what I mean, though. Is it just homology that matters?
that's what it appears.
Yeah.
And I mean,
And
in
also,
order,
in relation to that, if you did an experiment moving, not euchromatic, but on the X, moving the rRNA just as you have moved the euchromatic along, if you moved it further from the centromere, would you get the same result?
okay, so Dr. Catula asked if we were to move the rDNA, that's the kind of the normal pairing and conjunction site between the X and Y, farther away from the centromere, would it still be able to pair? I think, yes, it would definitely still be able to pair. We showed that all the homologies that we tested were able to pair, but what I would imagine would happen would be the segregation would not happen as efficiently.
So it would be similar to what you saw with your euchromatic.
Right, because again, it's satisfying the metaphase checkpoint. So whenever the pairing homology is farther away from the centromere, you can't feel that tension as well. So it's like it doesn't have the time to resolve, and metaphase will sometimes go on into anaphase without being resolved. And so those chromosomes can either become attached and both go here, or can come attached and both go there. So yeah, we weren't able to find anything like specific sequence that was special. I mean, the smallest one that we did was 120 kb. But they all were, we're still able to pair and segregate, which is really interesting. So it could just be any homology in general, or there could be specific little sites in there that aren't narrowed down yet. And again, like we mentioned, there's also could be potential for anti-pairing factors. So some areas.
Since we don't have a large audience here, and it's mostly committee members, I think we'll reserve the rest of the questions for Chris's exam.
All right.
All right.
I see you have very interesting data. How much of this data is transferable to other organisms, say humans, for example?
Well, so now that we've shown that any homologies can pair, I mean, pairing homologies is kind of, the Drosophila male meiotic system's always kind of been looked at as different because, right, it doesn't have the recombination. Oh, I forgot to say what the question was. Say it again, Dr. Asina.
So your study is based on a model system.
Right. So he's saying, how can we extrapolate what we've learned here with the flies to other systems? Well, now that we showed that any sequences can pair, people have always kind of said, well, the male fly is unique because it has these certain pairing sites, and these chromosomes come in, proteins come in and bind, and there's no segregation. But what we show now is even more viable. But again, it's really hard to study meiosis in humans. Females carry eggs all through life, and and a lot of things have been shown, or environmental aspects have been shown to affect that. But again, the main reason why this is important is it's just because of the aneuploidy and the dire effects that those can have on humans. And so this is just a model now that we can use and to better characterize what's going on in meiosis to kind of maybe help prevent in the future issues related to causes of meiotic mutations. All right, thanks guys. Thank you everybody at home. Thanks. Thanks for coming.
Thanks, Lee.
No problem. Thank you. Stop that. Oh. Hi. How you guys?
That was a mouthful, huh?
Yeah.